Shaw. We'll hear about the gut-brain connection in Parkinson's disease. We'll hear that will be from Dr. Poston. We'll hear from Dr. Ng about the Give Back Committee that's been organized, and it's sort of a work in progress, but I think we're sort of well towards achieving our initial goals there. We'll hear from Dr. Beth Mormino about imaging in relationship to aging, dementia, and what it tells us about our brain. And without that echo, we'll end up having lunch and then the World Cafe event that you'll hear more about. So Nisha, Dr. Scary, do you want to come up and tell us what else we need to know?
Okay. So again, I'd also like to join in welcoming all of you. Are we good with the online folks now? Can they hear okay? Lovely. So thank you all for your patience, people online. We really appreciate every single one of you and all your contributions, as well as those who could not join us today. We hold this hopefully annually, and we switched to springtime, just so you know, because we thought the weather's nicer and it's not close to the holidays. So that's why we made that change. In terms of housekeeping, many of you know already, but out through those doors, out through the second set of doors, and to the left, past the elevators, just past the elevators to the right are the restrooms. We also have volunteers. Oh, by the way, the outer door locks. So if you can come back through the other glass door that has a sign for ADRC and come around, in case you can't get back in through those double doors, we'd really appreciate it. If you have cell phones, please put them on silent now so that we don't have interruptions. And there are index cards on the tables. We will actually, there's a little deviation from what Dr. Henderson put on the screen. At 11 o'clock, we will have a short break for Q&A. And you're welcome to get up and walk around, but due to early departures by our two speakers, Dr. Shaw and Poston, we went ahead and made that a QA session so that if you have pressing questions directly, you can address those then. Go ahead and put your questions on index cards. We will have Samantha and Hillary doing the QA sessions, but there might be other people coming around to collect the questions on the index cards. And then lastly, I'd just like to take a moment to acknowledge all the volunteers, and also there will be those that can help direct you to the restrooms or upstairs if needed. They have badges working, and this is almost like Fort Knox here, so you'll need to get in everywhere. I'd like to acknowledge first and foremost Jamie for all her invaluable help in helping me get this coordinated and set up for today. But then also we have Jenny and Larissa taking care of the Zoom questions, Nayeli and Henry and Mackenzie and Lonja, we're doing registration out front. And I want to, oh, Hillary, of course. There she is, right there. She's waving. Samantha, why don't you wave so people know that you're there as well? So couldn't do it without the help of all these wonderful individuals. And without further ado, I'd like to pass it on to Dr. Henderson, but I'll be around if you have questions. Happy to field them. Take care.
Thank you. So let me just tell you sort of where we are to give an overview of the Stanford ADRC. This was a center that was funded in, I'm getting hand signals from the audience. Well, this microphone is not on. No, it's right here. So maybe I should just sort of, this man is in charge. He'll make sure things work pretty well, I think. But yeah, so thank you. I'm sorry my voice wasn't carrying as well as was needed. Thanks for that. Our center was funded by the National Institute on Aging in 2015. It's one of about 30 ADRCs now nationwide at different universities. And the goal is the same for all of these, and that is to provide an infrastructure for research into disorders that affect aging, cognition during healthy aging, enduring neurodegenerative disorders that occur during older age, particularly Alzheimer's disease, but not exclusively Alzheimer's disease. Some of the centers have been around a lot longer than ours. There's some that are more recent than ours. And so we're sort of in the middle of that larger pack. That said, each center does some things differently. Our center has focused on patients who have Parkinson's disease, Lewy body, in addition to Alzheimer's disease. There are a number of reasons for doing that. One is the expertise here at Stanford. Another is it's a really common neurodegenerative disorder that sometimes affects cognition, not necessarily all the time. And it provides sort of a comparison, a means of comparing and contrasting and seeing whether findings in one neurodegenerative disorder will carry over to another one, and if there are differences, what that might tell us about underlying mechanisms, and if there are commonalities, what that might tell us about prevention strategies or treatment strategies. We also are a center that's focused a lot on what we call deep phenotyping. That means collecting a lot of information from each of our volunteers. That's hard. It's hard on our staff, but it's even harder on people that volunteer. We ask them to come in and not only be examined and have their memory tested and fill out questionnaires, but also to give blood and to have their brain imaged, and in some circumstances to have their spinal fluid examined, and to give stool and to give urine, and to wear an actigraph to see how the sleep is, and to come back in a year and do some of this over again, and eventually give us their brain at the time of death. That's a lot. And we're just amazed that people do that. But it's also incredibly important because having this information aggregated, having a lot of information from an individual person, it's much more valuable than having one piece from one person and another piece from another person and a third piece from someone else. All this is valuable, but it becomes much more than just simply additive by having these longitudinal data. And so thank you, all of you for sticking with this. It's been really important. On this graph I'm showing here, you can see the numbers go up. This is cumulative enrollment. So by definition, these numbers never go down. We're up to over 700. Some of the older centers are higher than this. Some of the newer centers less than that. And you'll see two plateaus there. One around the year 2020 that was related to COVID, when everyone locked down, no one was coming in person. We're doing some things virtually then, and enrollment flattened off. And there's another flattening near the top of the graph, and that's when the NIH was in other government offices as well, was sort of in a turmoil. Funding wasn't clear. It wasn't clear whether centers were refunded, when we were funded, when the money would actually come in. And because we really didn't have funds in hand, there was sort of a curtailment of activities. We're really fortunate at that point to fall back on endowment income that, hi, welcome. And if I'm not loud enough, then let me know. From the Farouk Jamal Foundation, we had endowment income that allowed us to get through this over a year funding hiatus. We curtailed activities, but we didn't have to lay off any of our really important staff. Faculty managed to get by, and so that was what enabled us to carry through. So we're really grateful for that as well. So that's cumulative enrollment. The actual number is with us. That number went up and up and up, and now it's sort of plateaued a little bit. We need to sort of bump it up again. This refers to people who enrolled, but the drop off is because some people die. Some people move away to other parts of the country because grandkids and children have decided Florida for some reason is a good place to be, and so they moved away. Or there are other commitments or other exigencies that come up, and we understand that people can't, even though the intentions initially might be to stay with us for 20 years. Sometimes they don't, and all centers have some withdrawals for that reason as well. So our current active cohort, people that have been with us and keep coming back, is about 416. We thought that 500 is where we would want to be over time. We thought that would give us sort of the numbers of participants and volunteers to look at some of the important research questions that we want to look at. And so that's the goal during this funding cycle, to sort of build back up to around 500 to maintain people in this longitudinal cohort to think creatively about ways we can use our resources here or contribute to the national effort and research related to these neurodegenerative disorders. Just in terms of how we're doing and how people are staying with us, this is a graph that comes from the National Coordinating Center. It shows the proportion of people continuing over time. Stanford has done really well in this regard. It surprises me considering how much we ask of people. And so I think that is a testament to our staff. It's a testament to people here who have volunteered and sort of understood what we're trying to do here. Even though there's been some fall off, we've actually done very well. We want to do a little bit better, and we keep trying to think of ways to make things a little bit better. Our community advisory board has been really helpful in that regard. So I want to sort of say two other things quickly. One is a little bit about what is Alzheimer's disease, and the other is a little bit about some of the new assessments that are coming up in the clinical core. So this is, at least in my screen, there it is. It was more distorted up here than I see in the screen behind me. This is Alois Alzheimer, the person for whom Alzheimer's disease is named. He worked in the early part of the 20th century. There weren't research grants back then. There wasn't an NIH. And so he funded his research the way other people of his era did, and that was through family wealth. He wasn't born into a rich family, so he did the next best thing, which was to marry a rich widow of one of his patients. And there is Alois Alzheimer with Frau Geisenheimer, who helped support his research in the laboratory. He described several patients who had fairly early onset dementia. He found at the time of their death and looking at the brains under the microscope that there were changes that really seemed remarkable to him. Some of these changes were visible because there were new techniques that had been developed that allowed him to see changes in the brain that other investigators before him hadn't. And he talked about the tangles, neurofibrillary tangles in the brain, and you can see one of the drawings that he made of what these fibrils look like inside nerve cells that were either living or dying, or died, as he put it. And he described these other structures that are now referred to as plaques that didn't occur within nerve cells, but between nerve cells. And you might be able to appreciate this sort of a circular body. There's sort of these are two different stains, sort of a central core within the plaques. And this would be sort of a modern depiction of the tangles and plaques that Alzheimer described. And you can see the tangles are within individual nerve cells. Those are the blackened areas there. And the plaques are sort of those larger round structures that you see there. And we now know that these structures have several abnormalities, but the primary abnormality characterizing the tangle is a normal protein called tau protein that's been modified in a way that allows it to cause these fibrils to damage. And we know that the protein in the center of plaques is something referred to as amyloid or beta amyloid, which is also part of a longer normal protein that's been fragmented in an abnormal way that allows this accumulation. So we can now think about Alzheimer's disease not necessarily something that's causing dementia and can be diagnosed only at the time of death, but we also know more about what the protein abnormalities are that constitute that. And that has led to different techniques for trying to look at these proteins in people who are still living. You'll hear more from Dr. Mormino later about what brain imaging can tell the brain. These are slides that, these are images that come from the Alzheimer's Disease Research Center here, from the imaging core that she leads, looking at one way of trying to identify beta amyloid in the brain. And I know that just looks like a fuzzy gray, maybe brain up in the upper right-hand corner, but actually it shows an amyloid accumulation, and some of the fuzziness is due to the amyloid actually being there. The tau PET scans, the other protein, are colored here. It's a little bit easier to see. It's the red that starts off in the lower part of the brain, the temporal lobes, and typically over time, and people would start there and progress further on. So we can see with these kinds of scans, these proteins or measure these proteins. And with these imaging, we can tell where in the brain these abnormalities are occurring. Another major advance is we can now detect these proteins in fluids. These fluids include spinal fluid that is obtained from an LP. This is extraordinarily valuable for research purposes, and we can measure different kinds of tau proteins and amyloid proteins and others as well. And also, even more recently, from blood, from plasma, different varieties of these tau proteins, also amyloid. And these tools are becoming really quite good for trying to make a diagnosis of whether or not the brain has abnormal accumulations of amyloid and tau, and whether or not there are Alzheimer-like changes going on. We can't tell where in the brain these changes are based on fluids, but fluids also provide a different kind of window. You can measure other proteins that might give clues. Here we're looking at machines that will give us 11,000 different proteins in blood or spinal fluid. And we don't know what all these changes mean, but in aggregate, that information becomes really valuable about looking at mechanisms, looking at differences between healthy aging, between Lewy body disease, between Alzheimer's disease, and so forth. And this can be done in spinal fluid, this can be done in plasma, immune responses can be looked at. And having these kinds of biomarkers, these kinds of research markers becomes extraordinarily important for us as well. It also means that we can now think about Alzheimer's disease as not being a disease causing dementia with certain microscopic changes, but we can also think about this as a biochemical disease. And in a research setting, people are starting to think about, well, maybe you don't need dementia if you have these biochemical changes and that can be measured. I think probably that's not a direction, at least in the clinical sphere, that I want to go in. But in terms of research, I think that's a really valuable perspective. And it becomes even more valuable once we have effective forms of prevention, effective treatments. And then this early biochemical or imaging identification will be crucially important because it'll be like having high cholesterol. They're drugs that really work and reduce stroke and heart disease risk. And we want those kinds of interventions for Alzheimer's disease, for Parkinson's disease, for other kinds of disorders that affect the brain during aging. So finally, I want to sort of mention within the clinical core, we're going to be doing some sensory evaluations. We've already started smell evaluations. We're going to be looking at hearing and vision. And there are a number of reasons. One is we can look at sensory functions in relation to disease mechanisms, in relation to how memory and cognition is doing, some of the other biomarkers that we have doing. We can look at these as indicators of people who are doing well, better than expected, worse than expected. And it also provides an opportunity to give back some of this information that's clinically relevant to our participants. So smell assessment has already been started, thanks to Dr. Poston. You'll hear more about that from her. If you look at the olfactory bulb in the bottom of the brain, they're actually nerve fibers that are almost in direct communication with the external world. And that's really the only part of the brain where this is possible in that kind of direct manner. If you look at the olfactory bulb in Alzheimer's disease, you'll see tangles there. If you look at it in Parkinson's disease, you'll see some of the Lewy body abnormalities. And so we think that smell can provide important clues as to what's going on in parts of the brain that are affected. For hearing, there's been a lot of recent work suggesting that hearing loss is associated with dementia and Alzheimer's risk. We're not sure whether it's causally related or indirectly related, but we think that's important to look at. And we'll be looking at pure tone thresholds, standard word identification thresholds, and sort of a new speech understanding and noise test that Dr. Matt Fitzgerald from the ENT department, who is the chief of audiology, a test that he has developed. And finally, I'll mention visual testing. We'll be looking at visual acuity, but also looking at something called optical coherence tomography, which enables people to look at the back of the eye to measure the thickness of the layers of the back that have nerves in them, retinal nerves that have fibers in them. And there are reasons to think that these will be clues to what's going on in these disorders. We have other research projects here that relate to these sensory findings. And so we're excited about being able to implement those. We'd hoped to have this in place this year, but with the funding delay, then it's sort of been pushed back a year, but we're ready to get that rolling as well. So for that, let me thank you for your time. Let me introduce our next speaker, who is Dr. Sharon Shaw. She is the Division Chief for Memory and Memory Disorders. She is the Associate Chair for Clinical Research. She runs the Clinical Trials Program and just been really important in that sphere.
Thank you. Good morning, everyone. Good morning. Hi. You can hear me, yes? You may not be able to see me, especially depending on where I stand. I'm really grateful to be here with you today because I want to start off, and usually this thank you slide is at the end of talks that I give, but I really want to share my appreciation for all that you do, whether you participate in the ADRC, whether you participate in clinical trials, whether you support someone who participates, or you just want to learn more. I'm really appreciative of your support and interest in what we do here. I wanted to start off by saying what your participation has led to thus far. And you may not be a bit familiar with all of these names that are listed up here, but aducanumab was one of the trials that we were part of, the phase 1B clinical trial, usually is just trying to see if it's safe. And that's to remove amyloid, those amyloid plaques that you heard Dr. Henderson talk about. That's one of the early trials that we said, hey, can we remove that amyloid plaque from the brain and see if that helps people? That trial looked safe. So the company Biogen went on to do a phase 3 clinical trial called Emerge that we were part of. There was another parallel one that we were not part of called Engage. And that led the success of that clinical trial. There's a lot of controversy. We don't need to talk about that. But that success led to a modified FDA approval. And then on the heels of that was another antibody targeting amyloid plaques called lecanemab. And that was called Clarity AD, a phase 3 clinical trial that we supported here. And that's now been FDA approved as one of the first fully approved clinical trial, or fully approved medicine for Alzheimer's disease. And then on the heels of that, we were also lucky to be part of another phase 3 clinical trial called donanemab. It was called Trailblazer ALZ 2, and that was approved in 2024. And those are for people who have mild stages of Alzheimer's disease. So it was very exciting. And as Dr. Henderson mentioned, this amyloid plaque builds up. And is that a biological diagnosis? Is it a clinical diagnosis? We know people have this amyloid plaque buildup 15 to 20 years before we even have symptoms. So now there are prevention trials ongoing. We're part of lecanemab A345, where we're using one of those drugs in people who are cognitively normal and see if we remove those amyloid plaques, does that prevent the disease, the symptoms at all? So it's very exciting. We'll have the results for those maybe in a couple of years. So I'll be excited to share that with you again. I'm sure you're going to hear it from the press, hopefully on the same time. So I just wanted to say thank you, because that's led to these amazing discoveries, but we're not done. And you'll have specialists here in the room that will say, these drugs aren't good enough. You know, these drugs aren't worth it. There are a lot of side effects. There's infusions related to it. So my work is not going to be done. I really want to support diversity for our clinical trials. And I'm not in finance, but I've heard the term diversifying your portfolio over and over again. So in all senses of the word diversity is what I want to do for our clinical trials to support everyone like you. And so one is sort of, let's think about the mechanism of the drug. Are there different ways that the drug works besides just targeting the amyloid or even the tau? Are there other ways? We want to make sure we're addressing disease modification. So that means that we're slowing down the disease, preventing the disease from happening even. But for people who already have the disease, we're still supporting trials for drugs that help the symptoms so that you feel better if you have the disease. I want to think about different types of diseases. In our ADRC, we're recruiting people who have healthy brains, people who have Alzheimer's, people who have Parkinson's disease, people who have Lewy body disease. We really want to think about all of these diseases. And I'll talk a little bit later about people who have multiple diseases also. And then we want to think about the stage of disease. So when we have symptoms is when we go to the doctor, we get the diagnosis. Maybe I'll stand here a little bit so people in the corner can see me. Yeah. And then we want to think about even prevention, but also for people who are in the advanced stages of disease, we often find like there's not a lot to offer. So I want to think about the spectrum of stage of disease. And then what may not really be as salient to you is that when we think about clinical trials, that a lot of it is sponsored by industry or the pharmaceutical company, and that's great. And they usually have the funding. But when we have ideas as the faculty here, we want to implement that because there's some great discoveries from the people in this room. So we want to think about PI initiated, meaning the faculty initiates those ideas. And I'm going to highlight a couple of those for you today. And then collaborations. A lot of times we recognize we do better when we work with others, people who have different opinions than ourselves. And then the true meaning of the word diversity in terms of demographics. I want our clinical trials for the people who participate. So the results, when we say this works, we know it works for you, for me, for people who look like me, for everybody. So whether you're a woman, whether you're younger or older, what does that mean? Right now, we've been so wonderful to get some of these clinical trials, but we want to make sure that it works for everybody. So just wanted to highlight a few completed trials in the past, maybe 6 to 9 months. Autonomy was using an anti-tau antibody. And if you were really listening closely to Dr. Henderson, that tau protein that forms the tangles are inside the neurons. And so unfortunately, that phase 2 clinical trial was not successful, but I'll highlight this pink one below, which is another tau targeting clinical trial called Celia. But that one was targeting not sort of the tangles with an antibody, but a mechanism where it's called an antisense oligonucleotide, ASO for short, but it's sort of modifying the recipe that the DNA makes and then modifying what it describes, that protein that it makes. So, and then we were part of a clinical trial using metformin, so a common diabetes drug. We'll find the results later this year, I hope. So we were excited for those who have participated in those. And this antisense oligonucleotide clinical trial, it's a phase 2 clinical trial that we just completed. There was a press release about this maybe 1 or 2 months ago. So I just thought I'd highlight that. I'm going to move around so that people can see me a little bit. I'm doing a little dancing. So this was a clinical trial where we gave this drug in 3 different doses or a placebo over 18 months. And usually a phase 2 clinical trial is really to see maybe if their dose is right, see if it's safe, maybe some markers of success, but not really to prove that it works yet. And a phase 3 trial is that if the phase 2 works, then it's proving that it works. So this is a phase 2 trial and looking at these different doses. Unfortunately, it did not meet its primary endpoint. So the primary goal of that was to see if it worked. So to me, in my mind, maybe that's not the best goal because it's really about safety. It's a smaller number study. But what it did find is that it reduced the tau as measured in the spinal fluid and as measured on a tau PET scan. I don't know the details. We're going to hear more at an upcoming conference in July. So next month at the Alzheimer's Association. I'm blocking your view, Sally. And then, but what they did also find is that there were cognitive changes that were slowed down at all doses of this drug, but best in the lowest dose. So more to come. That's all that it said in the press release. The safety was good. It was similar to the prior study. And there's an ongoing long-term extension for the study. So meaning we'll learn more about safety. So that's exciting because all of the tau antibody trials have failed thus far, whereas this one is working a different way. These are all of our active clinical trials right now. Now, this is actually smaller because I removed some of the completed ones that just happened. We usually have about 10 to 15, but we have a lot coming in the pipeline. And I think what's changing is that we're doing a lot more PI-initiated trials. And I wanted to highlight a couple here that's in red to tell you a little bit more about them, because these trials are related to a faculty member saying, hey, I have an idea. Can we bring this to our patients? So this is a phase 2 clinical trial using a blood pressure medication called bumetanide, and that's led by one of our faculty members, Kian Yunus. And what some of the early data had shown, or the research that had shown, is that when this drug, because it's approved for blood pressure, was given to people, and if we looked at a Stanford database, a UCSF database, and a Mount Sinai database, those who were given this drug, bumetanide, they had an association of less likely to develop Alzheimer's disease. Well, that's interesting. And then what we saw is that this drug also, when we screened, or the researchers screened 15,000 FDA-approved drugs, this specific drug changed sort of the genetic recipe from Alzheimer's-like, which is what you see in the color code here, to something less like Alzheimer's disease. So Dr. Yunus said, hey, let's try this in a clinical trial. And through the support of the 9 Initiative and the ADRC, the goal is to recruit 53 participants who have Alzheimer's disease and give bumetanide for 6 months with 2 different doses to see which is the best dose. Because the earlier phase trial to see what's best here. It's not for blood pressure control, it's for treating Alzheimer's disease. So we're still recruiting for that. If that's interesting to you, Lanja is somewhere around in the corner. Yeah, there you are. Let her know that you want to hear more about that. Now, a different clinical trial that's interesting and has received some press is using a common drug for erectile dysfunction. Now, that's called a phosphodiesterase 5 inhibitor. That class of drug was interesting because in a UK database, when people were exposed to this type of drug, they were less likely to develop Alzheimer's disease also. And we can see in this graph on the left side is when people tended to favor the use of that drug. That class of drug helped reduce that incidence. And if you were 70 or older, it also was more favorable for the use of that drug. And if people had high blood pressure and diabetes, it was also favorable. And more prescriptions for that drug was favorable in reducing that risk of developing Alzheimer's disease. And why might that be the case? Well, it helps with perfusion. This drug works in multiple ways, this class of drug. So it increases perfusion, meaning blood flow, and that's what we think about erectile dysfunction. It's also used for pulmonary hypertension, so lung high blood pressure. But it also works by reducing inflammation and potentially reducing tau and neuroprotection. So there's an ongoing phase 3 clinical is that we've done a good job in figuring out whether people have Alzheimer's disease biomarkers, meaning like with blood tests, spinal fluid, PET scans, because about 15 years ago, we were conducting clinical trials in which we thought they had Alzheimer's disease, and when we did the tests, a third of them didn't have Alzheimer's disease. So we're doing better. So we're recruiting. That's why if you do participate in clinical trial or any research, we tend to do these tests to figure out that there's amyloid in the brain and or tau in the brain. And so that's great. But what we're recognizing is that as we get older, we're more likely to have multiple diseases in our brain. So just like we get older, we tend to have high blood pressure and high cholesterol. We tend to have multiple diseases in the brain. So the one pathology is not the rule. It tends to be the exception. So we've done a great job, but Alzheimer's disease alone is pretty rare. So how can we do better? And because our center tends to focus on that overlap between Parkinson's disease, sometimes we kind of use it interchangeably with Lewy body disease and Alzheimer's disease, how can we do better for clinical trials? So this is really to show that there's some researchers that took a look at the brains of people that had Alzheimer's disease and looked and saw how many of those brains also had the Lewy bodies. Those are the proteins that build up, or alpha-synuclein is a protein that form Lewy bodies and build up in the brain of people with Parkinson's disease and Lewy body disease. And about 40 to 55% of people who have Alzheimer's disease also had Lewy body. And when we look at Lewy body disease brains, 50 to 80% also had amyloid, the one you see in Alzheimer's disease. So we need to do better. How are we diagnosing this? And I don't want to steal anyone else's thunder, but Beth Marmino's group and others at Stanford looked at our own ADRC and said, when we look at people who have Lewy body disease with the clinical diagnosis, yeah, 50% also have amyloid. And then it was shown another way. It's very similar to what I showed you in that pathology study, where if you look at the Alzheimer's disease, just in spinal fluid, it was about 20% that also had Lewy body. And if we look at the Lewy body patients, about 50, 60% had also amyloid. Detect whether wearables and remote devices can be better to understand the progression of disease. And we're looking for healthy controls too. So if you're interested, to Alondra in the back. But what I'm highlighting here in red are leadership here. The ADRC at Stanford has really helped support the reputation and the amazing research that we're doing at Stanford. Beth Mormino, you're going to hear from her later today. She's leading the Stanford Aging and Memory Study for people who are cognitively normal, and a nationwide research study to really show how we can make sure that the imaging, whether it's PET scans, MRIs, can be standardized to better learn and be shared across the country and internationally. So I spoke too long. Usually I talk really fast. It's because I think I was moving around. Without you, we could not have done all of the work that we've done, including development of new therapies for Alzheimer's disease, but we want to do more. We want to have more accessible medications that are easy for everybody, therapies for other diseases, and therapies that represent you. Thank you so much. Now it's my pleasure to introduce Dr. Kathleen Poston. She's the chief of the movement disorders and really my partner in crime with everything that I've done here. Thank you. Yes.
Okay, perfect. I got that, yep. And you will have a chance to ask Dr. Shaw questions after, I believe, this session. So let's get this up. Oh, can I switch yeah. There we go. Okay, perfect. All right, so we're switching gears a little bit here, but you will see some very important overlap with some of the themes that Dr. Shaw mentioned with sort of thinking about very holistically this idea of neurodegenerative diseases. It's one of the things that I think our ADRC has done such a good job with, is really thinking very broadly, again, not just including people with a traditional Alzheimer's diagnosis, but across the Lewy body and movement disorder spectrum, but also thinking across movement, cognition, and now the GI tract. So this is, as Dr. Shaw said, I'm the division chief for the Movement Disorders Division. This is my fabulous faculty group. Yeah, so why are a bunch of movement disorder specialists here at a ADRC and talking about all of these things? And specifically, why am I a non-motor movement disorder specialist? Why am I spend so much of my time on the non-motor symptoms? So we're going to dig into that a little bit. And the background behind that is, when you think about the number of people who are affected by Parkinson's disease worldwide, we think about 6 million plus. However, the experience of living with Parkinson's over the course of a lifetime is very unique to individuals. One of the phrases that's often said by folks at the Michael J. Fox Foundation is, if you've met one person with Parkinson's disease, you have met one person with Parkinson's disease. And it really is the wealth of all these different experiences that patients have that impact their day-to-day life. because as symptoms progress, they vary from person to person, and neither, unfortunately, patients nor their doctors can 100% predict what's going to happen. But we do see these broad paths of similarities by observing individuals, and we can see certain patterns, but excuse me, those patterns don't necessarily occur in every person. So when we think about the spectrum of symptoms that occur, and again, I'm going to put Parkinson's disease and dementia with Lewy bodies kind of under this Lewy body umbrella, and we think about kind of on the top half of this graph, the motor symptoms, and the bottom half, the non-motor. And traditionally, what's in this red square are the things that we focus on in the clinical setting. But as we've learned more and more, we've started to learn about little things that happen years to decades before someone actually has the diagnosis of Parkinson's disease and dementia with Lewy bodies. Things like excessive sleepiness, anxiety, loss of smell, depression, and of course, constipation. And constipation has been observed for a very, very long time as being one of the very early symptoms that can occur in individuals with either Parkinson's disease or dementia with Lewy bodies. But it's been very hard to tease out because it's also very common in normal aging. And so how do you tease apart someone who's in their 50s who's having new onset severe constipation? Is that part of a neurodegenerative disease or is it not? And that's something that we are trying to figure out in our studies. So with that in mind, this idea that new onset GI symptoms later in life can be an initial symptom of a neurodegenerative disease, there's this kind of concept that friends of mine across the country and I have been thinking about, and could we possibly diagnose people with Parkinson's disease before they develop any motor symptoms on something like a colonoscopy, right? Like people start getting colonoscopies in their early 40s, mid 40s for some people. This is a routine thing that's done. Are there biomarkers? Are there things that we can start to look at? This is not true yet. We cannot do this today. I will be very definitive about that. But this is a vision that many of us have because we do see this early constipation symptom. It also has implications on treatments and what's really causing Parkinson's disease and dementia with Lewy bodies. So I'm going to walk you through that a little bit. Okay, so we talk about constipation, but in fact, the changes in the GI tract that occur in individuals with Parkinson's and dementia with Lewy bodies really affect the entire GI tract. It can be things like excessive saliva, we call that sialorrhea, difficulty swallowing, which is called dysphagia. People can have esophageal problems. They can have reduced gastric motility, so food just kind of sits in the stomach and doesn't go through very fast. They can have small intestinal bacterial overgrowth, and then they can also have things like constipation or incomplete evacuation. And so it really can affect the entirety of that tract. We tend to break it down into sort of the 4 areas of drooling, dysphagia, again, that swallowing problem, dyspepsia and gastroparesis, that's the slowing of the of the upper GI tract, the stomach and the small intestine, where it's just not moving things along very fast. And then constipation, the challenges with evacuating bowel movements. And we have a variety of therapies that we intervene with this, but we really don't have anything that's special for Parkinson's. We basically treat people the way anyone who had these symptoms would have. And so we don't have really disease-specific interventions for something that, quite frankly, really impacts people's lives quite dramatically. In addition, it brings up this question of what is causing these symptoms? So we know that the Lewy bodies in the brain are the classic finding on pathology for someone who has Parkinson's disease or dementia with Lewy bodies. And we know it's a brain disease, and so could it be these changes in the brain are impacting what's happening in the GI tract? Or could actually what's happening in the GI tract be affecting the brain? And it's an open question, I'll tell you right now. I have colleagues who are on both sides of this debate and very passionately on both sides of this debate. And I think it's still an open question. And that means it's something that we need to explore and understand better. And so why do people have this idea? Why do people think that what's happening in the GI tract could actually be affecting the brain? So the gut actually has billions of neurons. I mean, it's really, it's quite amazing how many neurons are actually in the entire GI tract. And a lot of neurotransmitters are actually produced in the GI tract. For instance, serotonin is 90% of the body's serotonin is produced in the GI tract. It's also a very immunologically active area. And that's in part because of all these microorganisms that live in our GI tract. We are actually more bugs than we are human, if you actually, from a cell count specific perspective. And so if you think about what's happening there, the purpose of the GI tract is to create a filter that allows for nutrients to come in while keeping out the things that we eat that you don't want to get into your bloodstream, right? So that's the idea. It's this barrier between what we're bringing in, helping the nutrients come in, but not having the rest of the world come into our system. Very densely populated, and so there's a lot of immune cells, right? Because if stuff gets in that your body doesn't want, we need to have a defense mechanism, an immune reaction. And when that goes awry, when you have this increase in inflammation, or maybe not enough inflammation in different areas, you can get what's called dysbiosis, which basically is a breakdown of that barrier. People use the word leaky gut. You probably heard that term. I will tell you, my GI colleagues do not like that word. They think it's not really specific and all that kind of stuff. But I think it helps me as a neurologist kind of wrap my head around what I'm thinking about with regards to the GI tract. Basically, you're letting in stuff that we shouldn't be letting in, and maybe even not letting in the nutrients that you should be letting in. So that's what happens when you get this breakdown in the GI tract. There's also a lot of communication. So again, there's the nerves that are communicating from the GI tract to the brain and back and forth. There's also these endocrines, so hormones and peptides that kind of, actually react when things are in the GI tract. And then you also have, again, this immune system that is communicating from the GI tract to the brain. And so it really is a two-directional communication between the brain and the GI tract. And so all of this together has led people to this question: why do people get Parkinson's disease? Well, we know as you get older, you're at higher risk. We know there are certain genetics that increase the risk, but the environment very clearly might play a very important role. And we know that neurodegenerative diseases are not exclusively brain-centric. As I said, there's a lot of things happening in the body, but there's some evidence that Parkinson's and dementia with Lewy bodies specifically, probably a little more so than Alzheimer's disease, might be caused in some way based on our experiences in the environment. And this comes from data that is actually decades old from a very wonderful and well-known neurologist, Dr. Bill Langston, who had this experience where he was actually in the emergency room here in the Palo Alto area, and met these individuals who had very suddenly, in their early 20s, had like sudden Parkinsonism. And it turns out that they had been creating some substances in their basement and accidentally created a substance that basically killed off their dopamine neurons very quickly. And then they investigated that, and turns out that substance is very similar to some pesticides that are used. And that created our first animal model for Parkinson's disease, where they would give that MPTP to animals, to mice, and then that kills off their dopamine neurons, and that's how we test drugs for Parkinson's disease. But it led to this idea that there could be things in the environment that are actually damaging dopamine neurons. So there's been decades and decades of work in this area. I will say this is still a hypothesis. We don't know for sure. We don't have 100% proof that exposures to the environment are the cause. One of the things that one of my dear colleagues, Carly Tanner, who worked very closely with Bill Langston for many years, the example she would say is that the genetics are like loading the gun and the environment pulls the trigger. You have to have that interaction between the two, and how much is genetics and how much is environment probably varies tremendously from person to person. And so that's kind of how we put it together. But we decided to take some opportunities here to integrate these and really ask some more detailed questions, try to understand this better. So a colleague of mine, Dr. Laren Becker in the gastroenterology department, combined forces with colleagues from engineering. We put this great team together of neurologists, gastroenterologists, neuroscientists, pathologists, And we applied for funding for this gut-brain Parkinson's disease consortium. If you want to hear more about this and other aspects, please go to the back table and see Dr. Mackenzie Bolin, who can tell you all about all of our gut-brain related studies that we are doing. But in short, this is a group of us from across the country. We all applied individually, and the NIH selected us to put together a protocol where we're all collecting the same things across all the sites to try to understand this connection between the gut brain and individuals with Parkinson's. We're going to be measuring everything from swallowing to colonoscopies to stool samples. We're really, head to toe doing the whole GI tract. It's quite involved, but I think it's going to tell us really comprehensively what is going on. We're also going to look at some very innovative wearable devices that are being developed in our engineers. And then we're also going to take actually samples from humans with Parkinson's and actually put them back into the mouse models to try to study some of this gut-brain interaction. And so really, at the end of the day, what our hypothesis is that this altered gut neuroimmune interactions, which maybe are mediated by some microbial factors, might promote disease progression and maybe even the cause of Parkinson's. So we're going to investigate this in detail in this study. And so this brings me back to my original question. What if we can diagnose and maybe even treat people at the time of something like colonoscopy or first evidence of constipation, if we really believe that there is this gut-brain connection? And so we don't know. This is something I'm going to spend the next couple of years of my life working on, maybe even longer. And I hope that it will be able to give us some insights, not just into the cause, but also potential opportunities for therapies for people with Parkinson's. So thank you all very much. And again, if you want to hear more, Mac will be here all morning. Thank you. And do I stay here? Okay.
So I'm gonna,
Sharon?
yeah, I'm going to ask Dr. Shaw to come up as well. And if you have questions, hopefully you've written them on the index cards and they'll come around and collect it. I'll start here.
Okay.
No, I think this is for Dr. Murray. Questions only for Dr. Shaw and Houston at this time, please. And then you'll address them yourself. We will have questions for Dr. Henderson, Moreno, and Ian before lunch.
Try and make sure that their question gets into their microphone so the folks online can hear.
Sure. Yes, absolutely. So for those of you online, we're just collecting questions right now. And we're happy to, if you guys have some and you want to give them to us, why don't you guys give them? We can go ahead and start. Yep. Yep. Okay. No questions. Sorry. Yeah. No questions so far. Yep.
Oh.
Yep. Yeah. If Hillary, if you've got any there, if you want to pass them over. Yeah. Okay. So while we're collecting some other questions, so regarding the gut-brain connection, is there a connection between celiac disease and Parkinsonism, specifically Lewy body disease? So there have been a couple of studies that have shown individuals with certain kinds of GI immune disorders. There's more evidence with inflammatory bowel syndrome or inflammatory bowel disease, but that has shown a slightly increased risk of getting Parkinson's. Interestingly, individuals who have inflammatory bowel disease and have been treated with some of the newer therapies, that seems to normalize the risk back to the general population. This is based on retrospective studies, so it has to be taken with a grain of salt. but that does make us think that there are certain inflammatory GI conditions that can potentially increase the risk. And while most of the studies have again been done in individuals with Parkinson's disease, there is reason to believe Lewy body dementia is also in that context, although there have been less studies that have specifically looked at individuals with Lewy body disease. This has driven several pharmaceutical companies to think about developing versions of those therapies that they are currently using for things like inflammatory bowel syndrome, and actually using them in early Parkinson's disease. There's and analogs of those. So there are some companies that are going into phase 1, phase 2 clinical trials looking and to look at that question because of this retrospective data that's been found.
Both of us. So I'll answer this first question. This person noticed with really good eyesight, semaglutide on one of my slides. Is this an ongoing trial? Is there any data? So there are 2 clinical trials using semaglutide called Evoke and Evoke Plus. We were not part of that clinical trial here at Stanford. The press and data that was presented both recently at the ADPD conference in Copenhagen earlier this spring, and in the fall, the Clinical Trials in Alzheimer's Disease conference last year, demonstrated that it was not successful. So this is for people who have Alzheimer's disease. That was Evoke, and Evoke Plus was people who have Alzheimer's disease and evidence of vascular disease or little strokes that have built up over the course of time in their brain. There was no difference. And it's really interesting when you look at the slides, when they show the people who were on placebo and then show the people who got the drug, they're almost exactly the same. So no benefit there. And people might say, why? It's been this magical drug for everything, right? We just heard it in the press. I heard on NPR the other day how this class of drugs, the GLP-1s, help reduce cancer risk. But what we see is that the data that have been looked at thus far about the association of these class of drugs and reducing the risk of Alzheimer's disease, it may not be causal, or you may have to use it in a prevention way as opposed to when people have symptoms. Maybe we have to go earlier. So there's still a question and there's still curiosity because these drugs have been so magical for people for so many different things.
With regards to, again, looking at diagnoses, so there is this skin biopsy test that can be done to identify Lewy bodies in the skin of people who have Parkinson's disease and dementia with Lewy bodies. And the question is, what about biopsies from the gut? Well, that's exactly what we're testing in the gut-brain connection study. So we will be asking for a skin biopsy from individuals and then also colonoscopy biopsies and looking at the concordance. And so that we are specifically asking that question to try to do this. And we are working with both of the companies that do both the cerebral spinal fluid synuclein testing and the skin testing as part of this to do that. And then also the causes and therapies, what about prevention? That's kind of my vision. I mean, again, maybe this is a Mars shot, I'm not sure. But if everyone in the US is getting colonoscopies anyway between the age of 40 and 45, what if we can just if we can detect the synuclein there before someone has any symptoms, or that maybe before there's anything in the brain, can we potentially prevent at that time? I mean, I don't think it's a crazy idea. I think it's a very hard idea to actualize, but that's exactly what I'm working on. So I hope to get there. And so stay tuned.
I mean, that's really exciting. I just have to comment before I answer these questions. It's really exciting, right? Because you're really changing the field. You and others in the room are really changing the field. Like I got a colonoscopy, all of us probably, not the younger people. And could we diagnose, that it's really impressive and amazing what you'll discover. Okay, my questions are, which Alzheimer's patients are not eligible for clinical trials? And then they have a subclause, specifically patients with greater than 4 brain bleeds. And so here's the conundrum for clinical trials is that every clinical trial is designed differently. A lot of them have similar sort of inclusion-exclusion criteria, meaning that oftentimes the people who design the clinical trials want to make sure that people are healthy, right? That when you try an experimental drug, we're not going to cause more problems when you take this. And if, for example, if someone has a cancer diagnosis and you're getting chemotherapy, they may not be the healthiest for this trial. And if something happens to them, then it could be attributed to the trial and the experimental drug. So what we've seen is that people who have more than 4 brain bleeds, or what could be associated with the diagnosis of what we call cerebral amyloid angiopathy, which is when you have amyloid not in the brain, but amyloid in the blood vessel, then those blood vessels are at risk for leaking more blood. So think of a leaky garden hose where you have holes in it, and then you could have more bleeds. And that could be just because of that disease as opposed to the drug. So in general, we want people who are relatively healthy or stable on medications, but every clinical trial is different. So if you're really interested in trying a clinical trial, there may be an opportunity. If you do have more than 4 brain bleeds, it depends on that trial. But in general, you're looking for people who are relatively stable in their other conditions. And then at what point would a person participate in clinical trials if you have a healthy aging brain participant? I love that question. So that's where in my slide where I'm talking about different stages of disease. So people have symptoms, that might be mild cognitive impairment or in that dementia stage with Alzheimer's disease or dementia with Lewy bodies or whichever, but we're starting to look more and more for prevention. So even with this gut, diagnosis, if we could figure that out, if you have a positive synuclein test, could we implement something? So what we're doing now, what we're seeing is that there are cognitively healthy or cognitively normal people that we're starting to recruit for clinical trials. The AHEAD 345 clinical trial where we're giving lecanemab. People are cognitively healthy, but they have amyloid in the brain. That's closed enrollment, but we're hoping to get some new ones on board very soon. So if you're interested, tell Londra. We'll keep your name on file, and when we have it ready for you, we'll tell you. Thanks.
And on that theme, if there, one of the questions, if I'm a healthy person, can I participate in gut-brain connection study? Yes, you can. We have some of our main study and some of our sub-studies. Please talk to Mac in the back. And then just a version of that of, is there any gut relationship being studied in people with IBS? It's been a little bit of a harder thing to specifically look at, actually, but you can talk to Dr. Bolin about that. Her PhD thesis was actually looking at colon biopsies from people with Parkinson's disease, inflammatory bowel syndrome, and individuals who had neither of those two. So she is actually the world's expert on that question. So please talk to her.
Okay, I have 3 index cards. I'm going to try to go quickly here. What is the neurocognitive data that removing brain amyloid and tau from the brain improves cognition? So I feel like it's planted from one of our experts here, like Dr. Grages or Dr. Henderson. So let me just qualify that at least with the clinical trials where lecanemab and donanemab got approved, it didn't improve cognition. So unfortunately, once the symptoms have started, what we're seeing that shows success for those clinical trials is that they're getting less worse over time than if you were not on the drug. And so what we see is there's a disease modification, so the slope changes. So if we're getting worse over time in cognition or independence, that with the treatment, you're changing that slope. It's not getting better, not necessarily staying the same, but you're changing that slope. We're hoping that someday we can keep it the same or even improve. So we're not at that point yet. But we do think that biology has something to do with it, that when you have amyloid and tau build up in the brain related to Alzheimer's disease, if we remove that or prevent it from building up, could that keep you healthy for longer? Any neurocognitive data from trontinemab cleared plaques in something brain tissue. So yeah, from the presentations at recent conferences, trontinemab is another monoclonal antibody targeting amyloid. And what's exciting about this is that it has been shown to remove the amyloid plaques in the brains of people who participate in a clinical trial. The problem is it was a really small study. So they need to do the phase 3 clinical trial called Frontier, which will be starting, or it's ongoing right now, but in the early stages. And the frequency for which you get the drug is less frequent than what we've seen for lecanemab and donanemab. So it's exciting, and there are less side effects. So if it does prove successful, then it would be probably better than what we have right now, but it still has risks and less feasible to take because it's not a pill. So that's exciting, but we're still in the early stages for that clinical trial. And then any trials on new drugs available who are E4, but pre-MCI or symptomatic. So I am not at liberty to say what when we're in discussions when we haven't signed a contract with a company but I will say that yes, there are some on the horizon. And hopefully we'll be recruiting for clinical trial related to this that have something to do with the genes and ApoE4, which is an increased risk for developing Alzheimer's disease, but maybe there's some association there with modifying either the gene or what ApoE4 does. And this is a question that maybe I'll open to you too, Kathleen. How is AI helping find solutions?
Wow. I think that the most immediate way is that one of the big breakthroughs that's happened in the medical field over the last 5-10 years is the amount of information that we can collect on individuals. So for instance, a vial of blood, we used to be able to send for like a handful of proteins. Now we can send like that tube of blood for 12,000 proteins. One of the things that we're going to be looking at in the PDGI study is this marker of transit time, where people are going to swallow this thing and it's just like a little pill, and then we take an x-ray and you can kind of see how far it is. Well, in the old days, the poor little radiologist would like have to sit there for hours counting all the different things. We now have an AI tool that's actually gonna look at all of that and be able to much more accurately tell all that information. And then also with like the wearable data, there's just so much data that comes out of that. So analyzing very high density data, particularly when you're getting thousands and thousands of data points on 40 people, statistically, that's a very ugly problem. But AI is actually really helping us to be able to do things that we could never do before with data. So that's actually the one that is actually happening right now that has completely revolutionized the way that we analyze data and making really just incredible strides there. I do hope that there will be more improvements in the way that we even think about collecting data and think about the type of data that we bring in. I think that that's been a little bit less clear exactly what that path forward, but there's a lot of hope with that in particular.
And I think in the clinical trials world, if AI could help design clinical trials more efficiently, find drugs, especially not new drugs, but even drugs that are around, really quickly assess for them. And then with the data that we collect from the clinical trial, could we recruit less patients or participants into them because there's so much rich data with the individual that joins?
Yeah, finding a way to fail fast is really important. These clinical trials take a very long time. And so if we could get to the answer that this doesn't work with 40 people instead of 400, that would be a huge step forward.
And then the other question, I think there may be an investor in the room. What are the top 3 public companies in this area? Lilly, any biotech companies? I'll just say that I don't know the answer to that. I'll just say that with lecanemab and donanemab, who have drugs on the market, that's Eisai, that's partnered with Biogen and Eli Lilly. And of course, I'm partnering with Lilly for the trial that I mentioned. But Roche and Genentech are in that major field with the question about trontinemab. Those are some major biotech companies, but there's so many that are emerging, and I wouldn't discount the smaller ones either, because they're looking for funding, right? And they really want to, they probably have something amazing, but they need the funding for that. So I don't know if you're trying to invest, but those are the ones that are available right now, yeah.
All right.
And please join me in thanking. Oh, I'm sorry. Oh, here. Yeah. Yeah, let me just. Thanks.
Thank you. Sorry
Hold your
to take
applause.
the time, but I just want to point out I'm a caregiver for my wife who's 11th year of the genetic dementia called Cadastile. Anyway, that with my support to her, and she's actually deteriorating faster in the last 6 months. And thanks to Dr. Gracious that also talking to Dr. Jia about the mobility, she's a mobility specialist. And also, I find that's definitely more relation with the to Dr. Paston's comment, the gut and brain. And I do see more issues. But again, the mobility issues and also the digestion and constipation and all these things. But I want to look at the alternative medicine as well, because I'm starting getting a little more short patience because I do see her deterioration and I want to slow down a bit. So I'm looking at the Chinese medicine a little more, and I do find there's some relationship about the whole system. In the Chinese have meridians, but I don't know much about it, but I do see some relationship. So somehow there is a talk about this relationship between the guts, the brain, the circulation, the qi, and all these things. So all I want to comment is I would be talking to maybe Dr. McKinsey. Anyway, a little bit more, and I like to also find out a bit more. And meantime, I would like to suggest maybe something in the future. There's some kind of collaborations by looking into the Chinese medicine as well.
So I think you brought up a really great point that, again, I think one of the take-home messages here is that it really is a whole body disease, both Parkinson's, dementia with Lewy bodies, Alzheimer's, and other causes of dementia, that it's not just the memory, it's not just the movement, and thinking about the whole body is very important. It's very important to us here at the ADRC. And thinking very broadly, I would just say the one comment I would make is that anyone who is taking or thinking about taking any non-prescribed medications, simply talk to your doctor about it. It's just really important that you keep that communication open and that they're aware of anything that you're trying or taking or thinking about taking, because if the rule of thumb is that if something is biologically active and it can help you, it can also hurt you. And it's just important to have that communication with your doctor. So that would be my advice to anybody. Thank you so much for those comments and bringing it back to sort of that whole body viewpoint, which I personally appreciate. So thank you very much. And Nusha, we're back to you.
Thank you. Yeah, join me in thanking them again. Appreciate it. Sorry. Okay, you're probably tired of sitting. Can we just take a really quick 2-3 minute, get up, stretch? Speaking of Chinese medicine, let's do breath work.
And I'm going to take my prerogative and make it 5 minutes to give people time to walk out and then come back.
Dr. Henderson. I'm the one. Kelly, just fine. Stop you right here and say, Dr. Henderson, don't say that. For your hand. Oh, well, I want to see you twice. Dr. I'm losing words. And it drives me crazy. Dr. Goulet sent me to you. Probably sent me to you because that's when you had me. If I were doing a study. Well, oh, yeah. Well, studying I'm having here, and my brain is. I still see the heck of it. If your computer dies, what do you do? So why did it die? All right, I'll get that. That's my wife's car. And I was up there when she got on ready.
Yeah.
Cool. Yes, well, I'm not, I have not studied. I do not know what you call, but anyway, I'm happy. Yeah. So far, no dragon.
Right there. Yep. Oh, God. Oh, it's a great escape. Yeah. Go ahead. Yeah, I was just gonna say it's great that the slide was here. Yeah. No. Right. So you and I have established how long ago?
Can we please reconvene? One thing, once we're done with our morning session, before you disperse for lunch, I have a few things to say about what the afternoon will be like. And then I'd also like to encourage I understand that there were some guests who may not have received a blanket in the bag. So if you did not receive one, we should have some extras. So you can check with Jamie or anybody in the back there, and they'll make sure you get one, okay? So I didn't want people to be left out in any way in that regard. So can we get people to take seats again? And I'll pass the mic to Dr. Henderson, who will introduce our next speaker.
So very quickly, thank you for being here this morning. Thank you for coming back. And I want to introduce Dr. Jessica Ng, who is a really important part of our Alzheimer's Disease Research Center. She plays a key role within the clinical core. She comes from the movement disorder division of the Department of Neurology, not the memory disorder part. So we're really pleased to have her here. And she's taken over responsibility for something that we refer to as a Give Back Committee, trying to think creatively about what kinds of data we collect, what kinds of information we have that might be useful to you, and to, in a small way, try to give back what we know to our participants and to the communities from which they come. So Dr. Ng.
Thank you, Dr. Henderson. So before I begin, I just want to thank all of you, our participants, families, and partners for your involvement in research, and what we do would not be possible without your time, your commitment, and your generosity. It's nice to see some familiar faces in the room. So one of the questions that we hear often from our participants is, what happens to the information I contribute, and will I ever hear about the results from my participation? And these are important questions, and this is truly at the heart of the work of our Give Back Committee. And today I would just like to share a few updates about what our committee has been working on, and in return, I'm going to ask your help in providing us some feedback on future directions for our committee. So why giving back? Historically, research has sort of been a one-way street. And at Stanford ADRC, we really want to make sure that our work feels like a true partnership. And the Give Back Committee is formed because we recognize the importance in shifting how we engage with our participants from that one-way street to the partnership. And the Give Back Committee was formed with several goals in mind. First and foremost is to acknowledge the tremendous contribution the participants have made to research. And then we also want to support our participant engagement by making research a more collaborative process. And lastly, we want to explore ways of returning personalized health information to our participants, and that information that people might find valuable. And when thinking about returning results in particular, we try to keep one central question in mind, which is, how can we share information in a way that is useful, understandable, and respectful? And to answer that question, we have brought together a multidisciplinary team. And it truly takes a team. The committee is co-led by myself, a movement disorder neurologist, as Dr. Henderson mentioned, and also Dr. Eunice, who's a behavioral neurologist. We also have bioethics expert, Dr. Taber, neuropsychologist, Dr. Rosen, who also co-leads Agree Dementia, which is the advisory group on risk evidence education for dementia. We have Denise Coley from our community advisory boards, as well as other faculty from ADRC cores, including Dr. Gracious, Dr. Romino, who you'll be hearing from soon, and Dr. Sholey. And the committee cannot run without our clinical nurse coordinator, Christina, as well as our research coordinator, Veronica. And each member brings a different perspective. And together we have spent many months discussing what information should be shared and how to present it clearly. As I mentioned previously, the work of the Give Back Committee truly represents the shift that is happening across research. And this figure just kind of illustrates how the participant engagement has changed over time. In the past, participants contribute the information, and they often receive limited feedback with little to no communication afterwards. And today, research is becoming increasingly more participant-centered. We emphasize transparency and are beginning to return the individualized research results when appropriate. Participants, I think of them as more engaging partners rather than just data sources. And looking ahead, our goal is to create an ongoing dialogue and partnership between participants and researchers, and we hope that participants can feel informed and connected to the research that we're doing. Of course, sharing results is not just sending information in the mail. It does come with some important responsibilities, and we want to be mindful of that. First, we want to make sure that the information is easy to understand. Us as physicians and researchers, we are, aware that we use a lot of medical jargons. So we want to make sure that, some of these research findings, which can be really complicated, are presented in a way that is clear, participant friendly, and sharing the results. And then second, we do want to explain what the results do and do not mean. A lot of times research findings are not diagnostic and need to be interpreted carefully. We also want to respect people's personal preferences. Some people want really detailed information while others prefer less. And of course, at the end of the day, we want to make sure that the results are useful and meaningful. And finally, we recognize that receiving health information can sometimes be emotional. So we want to make sure that we provide explanations and support for participants and families as well. And with these principles in mind, I'll walk you through how the process is going to work. So the process starts when the participant decides whether they would like to receive research results. For some people, they want as much information as possible, and while others prefer to not receive certain results. I think both choices are reasonable, and participants can decide what's right for them. And then the next step you're all very well familiar with, where you'll complete interviews, cognitive testing, blood tests, imaging, and other procedures. And after the research visits, there's actually an important step that happens behind the scene, where our research team meets together in what we call a consensus meeting. And this includes experts from different backgrounds, where we go over the information that are collected during the study. After review the results at our consensus meeting, then we'll put together a letter that summarizes the results for you, which I'll go over in more detail shortly. And after you receive that letter, our clinical nurse coordinator will reach out and call you to help you answer any questions that you might have, as well as connect you with resources if that was needed. There is also an option to discuss the results with an ADRC clinician, but we do encourage you to first talk about these results with your neurologist or your primary care doctor. So without further ado, what exactly are going to go into these letters? In the initial rollout phase, the letter will include several different types of information. First is the research consensus diagnosis, which is a diagnosis that the research team determined after reviewing the available results at our consensus meeting. And then we'll also provide vital signs on the day of the clinical visit, which includes blood pressures that are taken both sitting and standing, what we call orthostatic vital signs, and as well as heart rate and other biometric data like your height, your weight, your body mass index, or BMI. Another important component that we're going to share is a cognitive testing results that will include a cognitive screening score, as well as summary of your neuropsychological testing results. And finally, we will, for participants who have gone through the imaging studies, we'll include amyloid PET scan results. I won't spend too much time going over the amyloid PET scan, since you've heard that from Dr. Shah and Dr. Henderson, and Dr. Mormino is more qualified than I am in explaining that to you. But just very briefly, as a recap, amyloid is a protein that can be found in people with Alzheimer's, and amyloid PET is a scan that allows us to see that amyloid buildup in the brain. So let me show you exactly a few examples of what the different parts of the letters look like. So here I'm showing you the blood pressure and heart rate on the day of the visit for a made-up participant. And these are measurements that I know a lot of people are familiar with, but we also want to provide some context in helping you understand what these numbers mean. We explain a little bit about how the blood pressure relates to brain health, as well as some general reference ranges for normal and abnormal findings. Again, the goal is not to diagnose a medical condition, but to provide some useful information that you might find helpful in discussing with your healthcare providers. Okay, and I know this is a busy slide. It's not meant for you to read the whole thing, but I just want to show you a little bit of how we formatted it and how the information will be presented. So this is one of the tests that our participants do, which is called the Montreal Cognitive Assessment, or MoCA for short. We will first provide a little bit of background information on what the test is, and then we give the individualized score. And then we explain what the score generally means to kind of help put it in context. So one of the things that we emphasize, and you'll see in the description, is that MoCA is a screening tool. right? So it's not a diagnosis. And there are a lot of different factors that can go into affecting people's performance. And that includes like sleep, stress, mood, medications, and your overall health. And, you know, for instance, here, like a lower score doesn't mean that someone has Alzheimer's or other type of, you know, neurological condition, but, you know, might indicate, you know, another discussion or other evaluations with your doctor. So here are, you know, how the results for the neuropsychological testing will be presented. You know, it gives you a little bit more information and a more detailed breakdown on the cognitive strength and weaknesses. I didn't include the background information here just because it would be way too long and too wordy. But you can see here that we kind of summarize like people's area of strength as well as weaknesses or graded difficulties. For instance, you know, a participant might have strength in memory and attention and having a little bit more difficulty in language and processing speed. Okay, and then for the amyloid PET scan, there are 3 possible results that you might receive. One, you know, first is like, you know, no evidence of amyloid buildup, which is the first one. And then, you know, second one is like there is evidence of amyloid buildup. And then the third one is this kind of inconclusive result that didn't really clearly show presence or absence of amyloid buildup. I think this is the part where communication is especially important because a scan without amyloid buildup doesn't really guarantee that someone will never develop memory problems. And likewise, a positive scan also doesn't mean that someone has Alzheimer's disease. And we spent a lot of effort as a committee to think about how to carefully explain these findings and what their limitations are, and particularly in that background information. So our goal, again, is to provide accurate information, but without causing unnecessary anxiety or stress. So everything I presented here today is a brief overview of a lot of conversation amounts our committee, but really the most important voice are yours. And as we begin to send out the letters later in the year, we want to make sure that they are helpful and meaningful to you. So your feedback, you know, is really important. We would really love to hear from you. I put down here some questions that will kind of help us and start to think about like what type of feedback we're looking for, but I'm also happy to discuss like other questions that you have. And these feedback will directly influence like how we can improve the letters and our process moving forward. And I know that we'll have some time after Dr. Mamina's talk for a Q&A session too. So with that said, thank you for your partnership, for making what we do possible. And then I'll let Dr. Mormino take it away and teach us a little bit more about the brain as well as what the imaging says about it. Okay.
She needs the microphone.
Yeah, I'll grab it. Just make sure. Yeah.
While we wait for Dr. Mormino, by the way, you'll be filling out a survey after the event too, and we've put a couple of questions there specific about the give back. So if you think of things later when you're filling out the survey, please put it in there. Thank you so much.
All right, thank you. Can you guys hear me? Hear me okay? Great. That's gonna think about it. So my name is Beth Romino. I lead the imaging core in our ADRC. Also this year stepped into the role of associate director, which it's been a great opportunity for me, and yeah, I've really been enjoying it. So I'm gonna kind of take a step back and kind of dive into the imaging that you guys are doing with us. Imaging over the years has kind of become a really core procedure. You know, it's been brought up in numerous contexts so far. It's really important for research, for the give back we're doing to participants, and for like the interpretation of each individual. So I think we're kind of at a point where it's really a core procedure, and it didn't used to, it used to be that way, at least across both PET and MRI. We're at the point where the vast majority of our volunteers have done a PET scan with us. We really started integrating this modality, and it wasn't until 2021, and we had a few scans before then. So big picture. So PET it's a type of imaging, a tool that we use. It involves a radioactive injection of a drug. The technology is able to detect this positron emission. There's two types of PET scans that we do in particular, but there's many types of PET scans out there in the world for different, for the detection of different diseases. We also do MRI, so it's a completely different technology. It also allows us to take pictures of the brain, but here it depends on us disrupting a magnetic field. And when we do that, we're able to capture different properties of the tissue. So we're able to look at things like neuronal and tissue loss and different other types of properties in the tissues themselves. PET allows us to look at an exact target in the brain. So the targets that we image are this amyloid plaque that you've heard a lot about, and also the tau aggregations in these tangles that we've also mentioned numerous times. So the MRI, so you guys know you have done a PET scan because it involves an injection. You know you have an MRI because it's super loud. What we actually do at Stanford is we actually combine these two technologies into one session. So these two tools have been pulled together in a PET MR system. So that's what we do through our ADRC. We do both at the same time, and we do it twice. So we do it once for amyloid PET. We also collect the MRI data, and we do it again for the tau side and collect additional MRI data. So this is my cue to pause and say thank you for doing this. This is not a fun procedure, and we know that. We also really stretch it out. We try to collect as much data from you as possible because we think it's important. So I really, really just appreciate the patience and the willingness to do this, not just once, but twice. And then now we're asking you to repeat it with us as well. So amyloid plaques, we've talked about this quite a bit. It's one of the hallmark definitions of Alzheimer's disease in terms of the pathology. It's spread throughout the brain, and we have been able to image this hallmark pathology now for almost 20 years, actually, from a research setting. This is what a typical negative scan looks like. It's a little different than the example Dr. Henderson showed earlier, which was in grayscale. So this is in color scale. The warmer colors reflect uptake. Here, the warmer colors are not on the edge of the brain. So this is kind of this off-target pattern. This would be considered an amyloid negative or not elevated scan, as we heard in that report. Here's what a positive scan looks like. So you can see the contrast. There's a lot of warm uptake pretty much everywhere in the brain. This is the typical kind of pattern of positivity. And again, we see it everywhere in the brain. So now the other scan that we've been doing are these tau PET scans. This captures the other hallmark feature of Alzheimer's disease. So these tau aggregations that form inside the neurons, these have a much more focal deposition. So unlike amyloid that's everywhere, these tend to begin very focally in the medial temporal lobe, and then they spread outward from there. So we can image this. So here's two example PET scans on the lower portion of this slide. One scan is a very low tau scan, so there's no warm colors there. And then the other scan is an example of where we might see these elevations in this tau protein. So again, it's very obvious, it's kind of from a signal standpoint, but quite different from the amyloid in that it has this more focal deposition pattern. And one of the main things we're interested for both in research and also in how to integrate this with clinical information is how does this focal presentation change over time? Because of PET, we're able to repeat these in the same person and understand the progression of this tau signal. And now that we're doing, both of these types of, PET scans, another important thing to do is to, integrate this information, within a person, and then also in the broader research, sense, to be able to understand, the different contributions of these two Alzheimer's disease relevant signals. So here's two example cases. Both actually have elevations in this amyloid scan. One case is elevated in tau, but the other isn't. So, what does this mean? How is this going to change over time? How does this align with the clinical presentations? These are questions that we're very interested in. So blood tests have been mentioned a number of times. Every couple months, there's an article, in the news about the successes and excitement around these blood tests for Alzheimer's disease, and it is quite incredible that now we can actually get a very reliable test of this kind of initial change for Alzheimer's disease. It typically corresponds really well with the outcomes we see with these amyloid PET scans. But one thing we are missing, is the spatial information, the time course of these changes. And that's something we're quite interested in our centers. We obviously, we collect blood as well. And the connection between these blood readouts of these Alzheimer's proteins with the spatial distribution that we're able to assess with these PET scans. So that's the PET. Those are the two PET scans that we're doing with you guys. We also collect actually a lot of MRI data. So we have about 2 hours worth of MRI data on all our participants because of these two sessions. We can measure things like structure. So I'm just going to show a couple examples there, but there's also connectivity measures we can look at. We can look at diffusion measures. We can look at flow measures. So we're doing a fair amount of multimodality assessment with these MRI sequences, and we have a lot of that data to sift through. So here's just a typical structural scan that we collect. Here we can see this in this participant. There's loss, and that's kind of in the black kind of areas around the cortex. You can see that kind of throughout many regions in the brain. The arrows are pointing to the medial temporal lobe and also to the cortex. However, some individuals will show more focal loss in that medial temporal lobe region and not in cortex, whereas some will actually show loss in the cortex and not the medial temporal lobe. So MRI allows us to kind of get into this heterogeneity, these differences across individuals, and allows us to better understand, what that might mean and how that fits into the clinical picture. I'm going to say one comment about functional MRI. So this isn't something we do as a core procedure on our ADRC, but many of you, actually, if you're co-enrolled in the Stanford ADRC memory study, we do this quite a bit in that study. You've also may have been recruited to participate in a sub-study with one of our faculty within the ADRC or collaborating faculty. And in that sense, we actually have you do a task in the MRI scanner. We ask you questions and we measure your brain activation while the scan is going on. And we're able to look at, here are the regions that are activated when you are remembering something. That's also a huge ask of participants, and it's pretty uncomfortable, but that's another technique that we're able to take advantage of with MRI scanning. So to summarize, so in our ADRC and in our affiliated studies with the ADRC, we do many, many types of imaging. We really have been focusing the last couple of years on PET MR imaging, looking at the amyloid and tau proteins in conjunction with these MRI measures and how does this change over time. We've had this standard protocol in place now for a number of years. So I think a lot of folks have had this experience and hopefully are willing to do the repeat imaging with us. And, the field is moving so rapidly. So, we just started with the amyloid and the tau PET a couple of years ago, and we already have our eye on the next round of, kind of advanced, more like pilot style studies. But in particular, we actually got probably the best high resolution scanner in the world here at Stanford a couple months ago. It's a 7T scanner compared to the 3T, traditional 3T scanner that's in place. And we'll hopefully be running some smaller studies to see if we can improve any of our detection efforts using this more advanced system. There's been some breakthroughs in the field with regard to PET imaging of alpha-synuclein and Lewy body, so Lewy body aggregations, which have been a huge gap in our field. We're starting to see the first promising scans. And so I in my view, I think we will start this in our center hopefully next year once we have some more confidence on some of these early studies that are starting to come out over this last year. And that, we've talked a lot about the co-occurrence and the relevance of both Alzheimer's disease and Lewy bodies. This would be a major step forward for us to be able to actually image both of these in this research setting. So I will end there, and thank you for your attention and for your participation. And I think we're going to move to questions. If there are some.
So now we'll have open Q&A for Drs. Henderson, Dr. Ng, and Dr. Mormino. And so please write your questions on the index cards. And Mackenzie, Samantha, Hillary will be coming around to collect those and pose the questions.
So give us one or two while you're collecting other cards.
Yeah, if you could put who is for in terms of speakers, that would be really helpful. Thank you.
Yeah, so I think this is a question about potentially either an incidental finding on an MRI. So the question is about white spots on an MRI that were detected. So this could be a couple things. It depends on the sequence. It also depends on the context, but there are these MRI sequences that can detect changes in various kind of vascular related measures. So white matter hyperintensities, there's a sequence that we use to look at that. That's actually a very common change that happens in both normal aging, but also can be a risk factor for some impairment. And then there's also these like small strokes that can be seen and things like that. So it's definitely possible that any of these features were detected for this case. The thing you need to do with any of these like incidental findings or any concern is to consult with your doctor. Obviously, a lot of the research sequences we do too are not actually optimized for the detection of these changes. So if it was in the context of a research scan, it would be really important to follow up and get an actual clinical scan to confirm what the finding actually was. But it's very common given all the age-related changes that we expect in the brains. That's there might be something that might be a little suspicious.
Take the next question. So this is a question about sharing the results with primary neurologist. So I think this is something that we're very cognizant of and try to think about like what type of results to share that are actionable and something that would actually make a management decision change, right? Like thinking of it also as a clinician perspective, like what type of results would actually be helpful for me to know as someone caring for, the person. So I think that's why we have kind of prepared this letter in a way that is easy for you to understand, but also a physical copy that you can actually bring to your neurologist. And this is kind of reflective of my own personal experience in my clinic where people always ask for kind of my notes and so that they can share with their other providers, and that's something that we're trying to do. I think a lot of these testing results, again, the field is moving as we have heard earlier today, and some of these testing findings, it's not clear whether that will lead to a direct management change, right? So I think that's why we're kind of being careful about all these information that we collected and then what to kind of make available to you and to your neurologist. And I kind of echo, I think in here they said that I think one of their visits, the tester mentioned that the tests are not diagnostic. And it's true. And in the sense that a test result alone doesn't really tell me about how the person is doing, right? Which is why as a neurologist in a clinical setting, I don't normally routinely, or I don't even have access to order like fMRI, right? I don't normally routinely order amyloid PET unless there's a clinical concern, right? So we really truly as a provider think about a full person instead of just a test score, instead of just a imaging result scan, right? But so it's a little bit hard to make a diagnosis or kind of make clinical judgments based on just one test alone. And that's something that we do want to emphasize too, and something that we make very important in the background information as well as the letters that we're going to provide.
And I think going forward, we're going to try to make, we're working with our data core, so we'll make information available longitudinally, hopefully have some graphics. We're thinking about maybe a booklet format, but the first step is the first step, and we're not ready to do the other things quite yet, but we hope we will be soon.
So here's another question about kind of the information and potentially in the give back. And I think this is very good feedback for us to think about. So, and this is actually not the first time somebody from the community has asked me about centilloid levels. I was impressed to get a question about centilloids. So centilloids are a continuous scale on that we can extract from the amyloid scan. So in our current report, we have these like 3 levels, like you're elevated or you're not elevated, or it's kind of intermediate. So, the reality is that, there actually is this, continuous, scale associated with these amyloid scans. And that's what the centilloid scale is intended to do. It runs from 0 to 100. I mean, 0 is the kind of negative. 100 is what would typically be found in somebody with Alzheimer's disease, dementia. And then we have everything in between. So we currently are not, that's not part of the report, but I think that's very good feedback. And in some centers, they do integrate kind of a quantitative level, in addition to the kind of the category level that's provided. So I think we're trying to balance, kind of too much information. And, sometimes they don't line up. You actually can have a high centilloid value, but the scan is actually negative, and that has to do with errors in the pipeline, or off-target binding or things like that. So it's not a simple mapping too. But that, I think it's an excellent suggestion. And there was also questions about white matter hyperintensities, which I just mentioned, and also the tau data too from the PET scan. So I mean, I think we're there's a lot of information from the imaging side that we need to think about how to pull that together, and if right now makes sense to do that or to wait.
So my stack keeps building up here, and I think Nusha wants me to start getting into it. So one of these is a 4-part question. I'm saving that one for later. And 2 that are very quick to answer. One is, with a simple answer, is Alzheimer's disease genetic? We have our geneticist here, Dr. Michael Gracious, but if I can just sort of start off with a short answer, and that is, we heard about the loaded gun and pulling the trigger. I think that most of us have a gun inside of us, but the genetic factors, except for early onset Alzheimer's disease, don't seem to be, the gun doesn't usually go off by itself. There are some genetic mutations where if someone gets that mutation, there's a 50-50, if someone gets that mutation from one of his or her 2 parents, then the likelihood is that person will get Alzheimer's disease in midlife or early older age. For other forms of Alzheimer's disease, the other genetic contributions seem to be not as important. The one you hear about a lot is one of the variations of a lipid transport protein called apolipoprotein E, where one of the variants increases risk a lot. It's not determinate. Some people with 1 or even 2 copies of the E4 allele don't develop Alzheimer's disease. Some people without any copies at all do. And otherwise, we think a lot of genes have sort of smaller aggregate contributions. Dr. Gracious and others are trying to sort that out. And there are a few, maybe other rare mutations that might also contribute. Do you want to provide all right, I'm getting a thumbs up and then too many more cards. The other one of them is, you've gathered information on 500 people, 732 actually. Do we share with other universities? Yes. We share with anyone who asks, any qualified investigator from another university, but also all the Alzheimer's centers, including ours, contribute standardized data to a national coordinating center that's in Seattle, Washington. They in turn make those data in aggregate across all 30 centers available to investigators. This is all paid for by your taxes, and it's meant to be available for other investigators. And we try even parts that aren't part of the mandated data set to share as widely as possible. So thanks for that question.
I'll try to answer Basically, I got a question about whether the results will be shared with control party. They will. So basically we talk about, healthy control as well as people with Parkinson's or, different type of cognitive impairments. Like we all discuss, all participants who join our study at the consensus meeting. So anyone that we discuss in the consensus meeting will receive a copy of the results in the letter format that I had discussed. And then another thing is there's questions about whether we'll be releasing or returning information on tau imaging, and then what about like blood test results? So those are very good questions. I think as I mentioned, like this is sort of the first step and the initial rollout. So there might be in the future like other testing results that will including A4E4, as Dr. Henderson had mentioned. It's just there are things in our protocol that are not in place right now. So in the initial rollout, and again, like these are kind of information that we selected in the first phase, but I do anticipate in the future that we'll provide kind of more data as it becomes, more actionable.
Yeah, so there's a question about claustrophobia. So for our participants, during the screening for doing any imaging with us, we do ask about this. And if somebody, it's hard to know how you're gonna, if you've never done an MRI scan, how you're gonna react. So it really depends on you if you would like to give it a try, if you feel you're kind of claustrophobic but you don't know. We will schedule this session, and if you get in there and you don't want to do it, we stop. I mean, at any point, even without claustrophobia, if you want to end the session early, that's that is what we do. So it really comes down to you, how comfortable you are giving it a try. But that is part of our screen. And if you tell us you're claustrophobic, we might suggest, maybe this isn't the right procedure for you. We can do we can shorten the session a little bit. So sometimes we do that, if somebody really wants to participate, but anticipates that issue. But that's something, it happens and it's okay. And it's also okay during the screening, to realize this might not be a good fit. I have another question about different tau scans and actually different. So actually we don't use antibodies for PET. Antibodies don't really cross the blood-brain barrier that well, but we use them. They're small molecules. They're actually very, very low weight, but there are a number of them. There's 4 different ligands that are used across PET scans. And the question is just about how do we bring those together? And so, yeah, there are multiple efforts underway to harmonize those. There's a study that's ongoing. We aren't a part of this study, but a larger study that's taking the same participant and scanning them with multiple different types of scans to be able to do that correlation. And so far the data looks pretty well correlated. There is some nuance with that, but those efforts are currently underway and quite important for clinical trials. Should I keep on going? You guys?
I have a bunch.
Okay, why don't you?
So I will tackle two of this bunch. One of these is, are people with Alzheimer's disease more likely to get other diseases? And I'll interpret this question as meaning, are they more likely to get other brain-related diseases? And I think the answer is yes. It's a really great question. We don't fully know the answer. I think, as Dr. Shah alluded to, if you look at the brains of people age 70, age 80, almost everyone will show, not absolutely everyone, but some changes related to Alzheimer's disease, some plaques, for example. And a lot of people will show some changes related to Lewy body disease and Parkinson's. And almost everyone will show some form of vascular disease or another. And there are other kinds of proteins that are also associated. So it's really hard to avoid some of these changes with age. And the question is, though, are these more likely to occur when one of the other problems like Alzheimer's disease is already there? And the answer may be yes, but we don't know for sure. There seem to be more people who have Lewy body changes and Alzheimer's disease changes together than one might predict just based on overall frequency considerations. Common, there should be some overlap, but there seems to be a little bit more overlap than that. If that's indeed the case, the question is why? What does that mean in terms of mechanism? What does that mean in terms of treatment? And there we don't know the answers. It implies that there might be some underlying factor that might be predisposing to do two quite different neurodegenerative pathways, or it might be the one induces some changes in the brain that then affects the pathway of the other one. And so we don't know that answer yet. The other question, is there any research in the role of mitochondria in terms of neurodegenerative disorders? And again, here the answer is yes. There's been a lot of research in looking at mitochondria. This is a little part of a cell that's involved in energy. It's sort of the powerhouse of a cell, including nerve cells. This has been looked at a lot in Alzheimer's disease. It's been looked at a lot in Parkinson's disease. And I think the answer there is there's something going on of interest, but it's not something that's really jumped out at us after several decades of research. It's pretty clear that oxidation and energy is important, but whether some of this is causative or just part of the overall process, other processes that are going on, we really don't have that answer yet either.
So there's a question about lumbar punctures and if whether or not that provides information that imaging doesn't provide. So actually, just like the plasma, so with the lumbar punctures, with the cerebral spinal fluid that we're able to look at, we also can measure. We talked about these protein panels with 11,000 plus proteins. So it's a kind of a similar opportunity. There is one newer test that actually, is a very robust marker of alpha-synuclein positivity. So it's done via the seed amplification assay. That's a test that can be run in cerebral spinal fluid and is very highly correlated with postmortem examination of Lewy bodies. So there actually is a test that we have on the biofluid side that's specific to cerebral spinal fluid. You can't measure that in plasma, and that we do not currently measure with imaging. So that's like one key example. And then just broadly, I would just emphasize again with imaging, we get the spatial location. We don't get that with any of the proteomics. Do you want to add anything on the lumbar puncture?
No, that's good. Thanks.
Should you do more? Or?
Oh, I have a lot more.
Let me do one. Actually, let me do one more. One question is about the size of the hippocampus and if we're looking at that over time. And so, with these MRI measures, that is one of the quite standard measures we get is volume of the hippocampus. We can also look at subfields within the hippocampus, and we sample that in every MRI session that we do. So we are able to look at those changes over time.
Yeah. So here's a 4-part question. How does the intake of excess sugar, processed carbohydrates, alcohol, and stress, how do these contribute to neurodegenerative disorders? So the answer is they probably do, and maybe in different ways to different disorders. So looking at sugar, maybe processed carbohydrates, sort of a simple answer, which I think is not the total answer, is that these are associated with diabetes, and diabetes is associated with vascular disease, and that seems to be a risk factor not only for vascular dementia, but also for Alzheimer's disease. Also within the brain, because a number of these changes accumulate with aging, anything you can do to reduce one level of pathology, like reducing vascular disease, lowers the threshold for cognitive impairment. And so we don't know for sure how to stop Lewy body alpha-synuclein pathologies. We don't really know for sure how to stop Alzheimer's pathologies, but we do know how to stop some of the vascular pathologies, and that's something we can do about. And so, so I do think excess sugar, maybe processed carbohydrates, might have some role in sort of at least through vascular mechanisms, probably other mechanisms as well. I'd also point out that processed carbohydrates, I read, tend to be during these processes tend to accumulate more, plastics, these microparticles that at least in animal studies and some now in human studies seem to be associated with inflammatory responses contributing to diseases in the heart and possibly the brain. Alcohol. Well, in the old days, it was said that unless you drank way too much, maybe a little bit's not so bad, and particularly red wine. And these were studies that came out of the Bordeaux region of France, and everyone was happy. And I think now the data are sort of turning a little bit in the other direction. It's not something you can study experimentally. I can't randomize everyone in this side of the room to have a glass of red wine after dinner, and everyone on this side for the next 5 years to have no alcohol whatsoever. You can't do that kind of trial. And so you need to rely on observational data, which can be sometimes quite good, sometimes not quite so good. And I think the current thinking is alcohol probably is not good for the brain, that the associations we thought we're seeing with 1 or 2 drinks at night, wine, were the people who had the time to buy wine and go to the tennis club on weekends and live in better parts of town and eat healthier diets. And so it's not clear that one extra glass of red wine every day is actually protective. And maybe at some level, it's not protective. I think at lower levels, I wouldn't worry about it too much, but at higher levels, then I would. Stress. Another hat I wear is that of an epidemiologist. I work with colleagues in Denmark, and they, us, and others have also looked at stress-related disorders in relation to dementia. This isn't proof of cause and effect, but people with stress disorder diagnoses, with anxiety diagnoses, are at higher risk for developing dementia later in life. In animal models, different kinds of stress seem to be related to some of the markers that we look at for Alzheimer's disease. But I'd also noted that people who have undergone remarkable stress, people who survived concentration camps, for example, I haven't seen good evidence that the risk of Alzheimer's disease is markedly elevated in those populations. So again, the association is there, cause and effect is a little bit harder to tease out.
So there's a question about studies connecting. I'm loud enough, I think. fMRI data and cognition. So yeah, the functional data I mentioned during the talk, and we don't collect that during our ADRC. We do. So another hat I wear is directing the Stanford Aging and Memory study, which is a cohort focused on baseline enrollment healthy individuals, and we in that study, we do a lot of fMRI of scanning to look at activation patterns during successful memory, and then we relate that to both cognitive assessments and behavioral assessments taken at the time of the scan, and then also over time as well. So that design is present across many cohorts, that are interested in these neural indices of cognitive processes. It's just not a focus in our ADRC. And then I have a couple of cards ask this, like what images would be kind of the best, to learn more, like what are we missing? And I think there's many targets, we're not capturing. PET it's a wonderful tool because it's so specific, but even the amyloid plaques themselves or these tau tangles, we don't understand, how that actually directly connects to the mechanisms that drive disease. So I think, if there were PET scans that looked at these, the mechanisms that were actually, driving this disease and that we could target, therapeutically, that would obviously be helpful. And then I'm also just really excited about what I think is coming in the near future. I mentioned on the summary slide is just alpha-synuclein PET imaging. So we've been missing, the ability to look at the spatial distribution of this pathology, and I think we'll learn a tremendous amount about disease progression once we have that implemented.
So a couple more questions. One is about autism, and a really important question, but one I don't really know the answer to. Autism is a really common diagnosis. It's becoming more common. Part of the question is there something triggering more autism in the population, or what's more likely? Are we just more aware of it and doctors and educators and parents are sort of making or driving the diagnosis when otherwise it would have been missed, or sort of detecting changes at an earlier stage or at a milder degree. But it's very common. I think one of the reasons why we don't know for sure whether it's related to disorders like Alzheimer's disease is that these tend to be childhood diagnoses, and knowing that someone has a disorder in childhood and then knowing what the outcome is going to be in 50 years takes time to sort of put that information together. And people are working in this because, again, it's a really common problem that affects the brain. And there may be deeper connections than I'm aware of, but that's sort of my current understanding of that. I have a question about exercise. One is a memory exercise. It's commonly said, I say this, that it's good to be active. It's good to come to conferences like this and try to stay awake and listen to the speakers. And on the way home, see if you can remember anything that we've said. We think that sort of builds brain reserve. It probably doesn't remove plaques and tangles from the brain, although I wish it did. And I don't think there's one particular kind of memory exercise that's better than others. If you're the kind of person that likes computerized games or computerized programs that help you develop mental skills, that's good. If you'd rather play a musical instrument, that's good. If you'd rather engage with grandchildren, really engage with them, get down to the floor and interact with them. That's good. That uses your mind. Playing cards. I don't know that there's one kind of memory or cognitive exercise that's better than others, but certainly all of these are better than watching Wheel of Fortune. I think, and the other question I think is in exercise more generally. I think it's really clear that if you're a mouse and you're put on a treadmill and you have a genetically modified predisposition to developing Alzheimer's-like changes, exercise is good. It does a lot of things that are really good for the brain. It increases vascularity, increases like something called BDNF that is associated with better brain function, maybe reduce Alzheimer's risk. It's associated with less degree of some of these Alzheimer-like changes. So again, if you're a mouse, ask for that treadmill. Let your provider know that. For humans, we have a lot of exercise studies looking at cognition. It's hard to do an exercise study and then look at Alzheimer's disease 15 years in the future. The observational studies in general tend to show that people who exercise more aerobic activity, also resistance training, have a lower risk of developing Alzheimer's disease. But that may be because these are the same people that drink a glass of red wine and eat better. There was one observational, not experimental, group coming out of Britain, a retirement community, looking at people who later, these are people followed for like 30 years, who later in life developed dementia. And people who developed dementia during the last decade before their diagnosis were exercising less than the people who didn't. But if you went back 30 years, they were exercising the same. And about 10 years before the separation occurred, there was a difference in exercise. So would you conclude that if only this one group had really kept with it and exercised more, they wouldn't have dementia? Maybe. Or were there earlier changes that affected motivation, sort of the get up and go, wanting to socialize, wanting to get out there and run, that reflected early pathological changes? We don't know. There are several. I'm still not getting the hook off the stage yet. There are several really large studies, a number of studies in different countries that are looking at intensive interventions in older people. I think these are really important studies to do. They're looking at people at higher risk, not with dementia, but higher risk by virtue of vascular disease, by virtue of poor diet, by virtue of other kinds of risk factors. And the interventions are exercise or mental activity. They're trying to take care of vascular risk factors. And then following these groups over time, the first group to report outcomes from Finland, they reported that over a 2-year period of time, this high intensity intervention group that included exercise showed a cognitive difference that benefited the active group, but the benefit was small. It was 0.02 of a standard deviation per year, which is statistically significant, hard to recognize an individual level after 10 years. Who knows? Those data aren't available yet. There's a similar study that just was reported out from the US, the US Pointer study, that showed exactly the same thing. There was 0.03 of a standard deviation difference per year in their 2-year studies. And there's similar studies going on in other countries. So I worry that exercise is going to have a huge impact when done for several years in older age. I think a lot of these lifestyle interventions are really important, but may need to be started earlier in life, in middle age and younger age, maybe even in childhood. And so that may be the end of my cards.
Can you please join me in thanking Drs. Henderson and Moreno one more time?
And if any of us have neglected any of your cards or not giving you the answer you wanted or a full answer that you expected, come grab us.
Or feel free to send questions to the ADRC Stanford at stanford.edu email that you all got. for me with all the information for today, and I'll be happy to pass it along to folks. I think we also did address the online question, so I don't think there's anything online. Just want to thank everyone for their attention, but before you get up, lunch will be served outside, but I have a couple of things if you could just humor me for a moment. Wanted to give you just a quick overview of what we'll be doing after lunch, and we'll probably reconvene around 1:05, so you'll have about 40 minutes to have the afternoon session. That'll be like a World Cafe style, where we'll have 4 breakout groups, one in this front corner, one in the back corner, one in that back corner, one in this front corner. Feel free to get into a group of about 20 folks thereabouts. We will have members of the Community Advisory Board, as well as our outreach, recruitment, and engagement team facilitating that. Nayeli will be waving her hand there. She'll be giving you the same overview again after lunch, when she'll give you a little bit more detail. And then I just want to thank all of you again for your attention and patience with the morning, but I hope you'll get some good sustenance out there.
Thank you all so much for engaging in thoughtful discussion and dialogue. What we'll do now is go around. I'll pass the mic to our mighty facilitators and note takers to do a quick recap, 2 minutes, 1 to 2 minute key takeaways from your discussions. So we'll start with group 1 on caregiver supports and resources.
Hi, everyone. My name is Henry. I'm with the OREC team here at the ADRC. I will start off by talking about some of the key takeaways from this particular station, which is on the topic of caregiver support and resources. One of the main takeaways that we did discuss here is kind of having considerations for caregivers that are sometimes forgotten or overlooked. This could include how to outsource for basic care and maintenance. Sometimes we don't consider what long care looks like, right? If I'm a caregiver and my time is tied up in providing care for a loved one, sometimes we don't have time for care in the sense of lawn care, meal preparation, house maintenance, right? All of our time is focused on providing care for our loved ones, and how do caregivers access additional support to do the other things that we don't necessarily have time to deal with. Another thing that we have as a takeaway is caregivers need support, such as where to begin, how to take care of someone who is ill, and then how they can create a space for themselves as they navigate the process of caregiving, right? And one of the main questions that does pop up is the idea of how do I take care of myself while I'm responsible for the care of a loved one, right? Sometimes we don't have those considerations for oneself. And that's one of the things that was reiterated across all groups. And then one of the last things that we did, just to synthesize some of the conversations that we did discuss, is caregivers need resources they can access, which can inform them about critical decisions that they will need to make on behalf of those that they are caring for, including estate planning, vetted resources for caregivers, power of attorney, financial planning, medical planning, and at-home support based on the needs of the patient, right? Sometimes we don't consider what those needs will be or how they will arise. And caregivers do need access to these resources in order to make informed decisions as it relates to care. So those are some of the points that came across and we kind of synthesized for our portion. So I will hand this off to the next group.
Thank you. We'll go with our group 2, which is ADRD communication and family dynamics.
So some of the takeaways are there's a mixture of experiences around comfort with talking about Alzheimer's and other dementias with family members and friends. Some people are comfortable talking about it, and other family members don't want to talk about it. Another thing is training and education of everyone involved in the care of a person with Alzheimer's is important for making the conversation easier. And also there was a comment about training for primary care physicians, because a lot of primary care doctors don't want to even tell the person their diagnosis. Question 3, people are getting resources from a lot of places, online, from friends, from family, from ADRC, from physicians, and from AI. And I want to remind everybody, the Alzheimer's Association offer a lot of information too. So you can go to the Alzheimer's Association website. A lot of interest in genetic testing. One of the resources that they were saying they need that they don't have is genetic testing. Some doctors won't even give access to genetic testing and to the results when they have the test, once they get it.
Thank you. Or topic number 3 on outreach, recruitment, and engagement.
Well, thank you all so much for the wonderful discussion. Our team asked you a little bit about how you heard about the ADRC, what brought you here, what motivated you. We heard many things. We heard that many of you learned about the ADRC by connecting with your own providers, whether it's your neurologist or a clinician. I think similar to the other group, we had many recommendations about how to increase that and formalize that. There's like trainings for others to know that we exist, so other neurologists and others can join us. There was a great recommendation, for example, to do an event similar to this, but geared for providers, so for those neurologists, those clinicians, so they know more about the ADRC and can refer. Many of you as well have a family history of some kind of dementia or similar condition, and that's a big motivator for many of you to join the ADRC, to continue participating every year, which we really appreciate. But also many of you expressed that because of that, you want to hear more about your results, your particular risk factors, your developments over time. So we heard loud and clear that you want more of that. I think it's a bit of the efforts that the ADRC is making around sharing results, but we heard that in many of the groups, so really appreciate that feedback as well. Many of you had some brilliant ideas for how we can let other community members know about the ADRC and the opportunities to participate. Several of you mentioned something similar to an ambassador model, where we can work with some of you, provide you maybe some training, provide you some materials, and help us recruit and disseminate the word in the community. This could be even with simple things like providing an email template, which we heard in this group, that you can like simply send to others that you know that might potentially be interested. I mentioned a little bit of like working more closely with neurologists, clinicians, individuals who work at memory clinics. We heard a bit of an interest in partnering more formally with other hospital systems. Not all of you are Stanford patients, so thinking about the other providers in our areas, Kaiser, PAMF, others, Valley Medical, where many people are receiving care, and that might not be as well connected with Stanford, and definitely not with our studies. And we also heard quite a bit about that there's a lot of stigma as well in the community, a lot of fears of participating in a study like this and learning more about yourself, your family history, etc. So a lot of more need also for education in the community, combating stigma, and also that for many individuals, they just simply don't know that the opportunity exists. So making sure doing better at disseminating that information. Thank you all so much.
Thank you, Ting and Patricia. Last but not least, Christina will tell us about return of results and research value.
Thank you. We, as Nayeli said, we were talking about research results. The clear theme was that participants really want their results to be data driven. They want to see the data. There was discussion about wanting some type of lever for themselves. Maybe some of them don't want everything, but they just want a little bit, enough to know what they can do, versus the data dump of wanting it all, and wanting some autonomy in determining what that might be for them and their families. Wanting the results, or wanting to participate, not just for their own selfish reasons of wanting to know what's going on with them, but also just contributing to their future, either their children, their grandchildren, or just future generations, protecting their family's health, and a desire to contribute to science. I would also say, in terms of the results, they want a path forward. So you all have said, it's not just getting the data, but what can I do with it? What are those actionable steps? The brain health, the activity, reducing stress, reducing our alcohol levels, to know what I need to do to best live out life. And then in terms of the data, again, there were some requests that for many of you who have been in the study for 10, some 11 years, that you'd love some of that historical data if that's able to be shared at some point. I think that mostly covered it. We did talk somewhat about the communication of the results, how people, how you all would like to see the data come back to you, or summaries, et cetera, of your work with the clinicians. All mostly wanted in writing so that you can reference that, so that you can share that with families. There was also discussion of phone calls versus emails versus letters versus texts, and how people want to get their communication shared. So I think that sums it up. Thank you.
Back to you, Nusha.
Dr. Henderson wanted to have a few words, and then I'm going to wrap up today's session for us.
So I guess we get another summing up. Mainly, I just want to thank everyone for being here. I wanted to thank Nusha and Jamie and others for helping organize this. And this has been really fun and exciting for me. My favorite part wasn't just listening to my fellow faculty who were presenting, and that was a treat for me, but also being able to answer questions and do it directly and not get repetitive use problems from trying to respond by email. So I really enjoyed the question and answer session, and thank you for those. Thanks again for all that you do for the ADRC, all that you do for other research programs. Help us find an effective means of prevention and cure, and we'll do it again next year. Nusha?
Sorry, I took pictures. All right.
Thank you.
And Nusha will give you some homework.
Yeah. So my first item for the day is, you all have something on your tables there that's got this, what's called a QR code, that you can point your phone over, and it should pop up something that can go into like a Chrome-like thing, and you can fill out that survey. It's really only, I think I want to say maybe 6 or 7 questions. If you prefer to do it on your computer at home, that's totally fine. I will be sending out a thank you for attending. Here's the survey link. Please fill it out in the next couple of weeks while the information is still fresh in your head, or if not, the next day. That would be even better. We really value your input. I hope you find that, based on the way the planning has gone over the years, that we definitely do pay attention to what you say, and we do our best to meet your needs and expectations in that regard. So kindly fill out the survey and give us that helpful feedback.
So this is a memory task. So you have to remember what you liked and let us know, so we can try to, bring something similar forward. But let us know what you didn't hear about. We can't do everything. What would you like to hear that you didn't hear? That's really important for all of us in planning.
Yes, thank you for that. Yeah, and then In addition, I just also checked with Nayeli. When I send that email out to all of you, I asked if they can give me the key takeaways from this afternoon World Cafe session as well, so that you would see that as well. And we'll also take it to the team and bring that up. I don't want to close without taking the opportunity once again to acknowledge Jamie Kramer, who really helped me put this entire thing together. So I really appreciate you, Jamie. Hillary, Jenny, Larissa, Lonja, Samantha. Jenny, Larissa. Wait, I'm trying to think. Who did I leave out? I know Nayeli, Henry. And if I left anybody out, I apologize.
This is your memory test.
Our community advisory board members, Leslie, Christina, Sandra, Ben and Ting, all of you, we really appreciate you. And who? Alyssa, yes, and Alyssa. One more thing though. We couldn't do this without all of you. So thank you so very much. You all deserve a round of applause. So yes, I'm hoping we will pretty much, sounds like we got positive reception for the spring. So we're going to keep it with the spring, probably not so much in June, but maybe a little bit earlier. We have to kind of go around different types of events that go on board. So thank you, everybody. Again, enjoy the rest of your afternoon. Keep safe, and we look forward to seeing you again soon. Be well.